everyone. Nice to meet you. Let me introduce myself again. My name is Femia. I'm from the Technical Supporting Group for ELISA Pipeline of Elabscience. It's my pleasure here to share you with my knowledge and experience on ELISA. Our topic today are mainly divided into three parts. In part one, I'll introduce you about our ELISA product lines, and in part two, we'll learn together about how to analyze and troubleshoot the abnormal results of ELISA. And part three will be a quiz time, and we also have gifts for you. Okay, now let's begin with the first part, the introduction about our ELISA products. At present, we have more than 4,500 ELISA and CLI kits covering many species like human, mouse, rat, etc. In order to better meet the needs of the market, we are constantly optimizing our products and launching new product series. There are all of the ELISA product lines we have now. The MS ELISA keys and high sensitive ELISA keys and uncoated ELISA keys are our new series. Here I listed some features about those different product lines. You can see the traditional ELISA keys have the most varieties for multiple species and multiple targets. And the Quickie Pro ELISA keys only needs 90 minutes and can complete it in just one step. The MS ELISA keys need the least sample amount as only 25 microliters. The high-sensitive ELISA keys can detect protein at femtogram level, and the uncoated ELISA keys would be your best choice to save on budget and build your own ELISA system. Now let's move on to the main part today, analysis and troubleshooting of abnormal ELISA results. During the presentation, please feel free to type your answers in the chat window, and we will answer them one by one later. In this part, we will talk about how to interpret an ELISA result and the possible causes for some common ELISA abnormal results. Okay, let's see this picture. This is a microwell plate for ELISA experiment. Every strip can be removed and saved for the next test. Usually, we would put the standards on the far left, and then the sample group 1, the sample group 2, etc. For accuracy, we recommend duplicate wells for both standards and samples. But if you want to save the strips or save the samples, or you just think it's not necessary, simple well is also fine. After several steps to get the data, we use Origin software to do the data processing. Of course, it won't be just this one choice. Common data processing software also can be used, except Excel. Then we paste the data into worksheet and use 4PL function to fit the standard curve. After we obtain the standard curve result, this Orange software can automatically calculate the sample concentration by just inputting the audio values. About the detailed operation, you can check the technical article on our website, and I have this link here. Okay, so how to quickly understand whether your ELISA result is qualified? Here, on the right side is an actual result of both standards and samples by sandwich method. First, we check the blank well OD values and the maximum OD values of standard curve. The blank well OD should be should be no more than 0.1, and the maximum OD value should be above 1.2. Otherwise, the standard curve would be unreliable. Then we check the then we check the sample OD values. They should fall within the range of standard curve. If not, maybe you should dilute the sample and test again. If the dilution ratio of the sample is too large, you can use stepwise dilution. Okay, if the OD values seems no problem, then we do the data processing. There are two important parameters should be noticed in this process. One of them is precision. It shows as CV% here. This value should be no more than 10%, since the lower this value, the higher the accuracy. And the other one is linearity. You can find it as R-square value in this table. The linearity should be no less than 0.99, so that we can trust the result calculated based on this standard curve. Okay, today I have chosen 5 common abnormal results in our daily ELISA experiment, and I will help you to know what could make them happen and how to avoid them. The number 1 is overall OD value is low or no signal. Here is one of the human interleukin-6 detection result for a serum sample. You can see the whole plate is colorless, and the reading data on the right side is basically around 0.06, which means no signal. Usually in this situation, we would first consider if there was any wrong operation or maybe a problem of kit performance. To analyze this problem, first step is to check whether the performance of HRP concentrate has changed. You can mix the substrate with HRP concentrate and see whether the color changed. Turning blue means normal. If it is normal, then it may be caused that the biotinylated detection antibody and HRP conjugates are used in wrong order. To confirm it, it's suggested to retry the experiment and see if it occurs again. If you use reagents from other kits, the result can also be no signal or low signal, because each our ELISA kit is optimized with its own reagents. Sometimes the plate is not completely colorless. It has signal, but very low. For this case, it has many possible causes, and I listed them here. Most of them are improper operation. Okay, let's move on to number 2. Overall OD value is positive. This is the opposite of the first one. The whole plate became yellow, even the blank well. And here is another result of the human interleukin-6 ELISA kit for serum samples. The overall OD values are over 2.0. This result is usually more relevant to the experimental operation. Since high and low OD value means different in sandwich ELISA and competitive ELISA, we would analyze this problem in 2 directions. For sandwich ELISA, we would first consider the contamination in the wash buffer, then the mismatch of reagents. If you use microplate washer, please make sure the machine tunnels are clean. For competitive ELISA, in addition to the above 2 cases of contamination and reagent mismatch, we also encounter cases that the customer just mistakenly used the sandwich ELISA operation steps to do the test with competitive ELISA kit. As we know, in competitive method, the sample and the detection antibodies should be added in one step. If they were added in 2 steps, the captured molecules wouldn't have antigen to compete, and it turns out the whole plate would be yellow. Okay, for this kind of for the number 3, all the values of standard curve is abnormal. For this kind of problem, the measurement would commonly appear as 3 cases. First, the standard curve dilution linearity is poor. In this case, we usually consider that it is because of the non-standard experimental operation, such as insufficient mixed standards, the inaccurate pipette volumes, and the bubbles in wells. For insufficient mixed standards, please ensure thoroughly mixing in each dilution step. And for inaccurate pipette volumes, please using calibrated pipettes and add samples vertically and quickly. And to avoid bubbles, please please centrifuge the working solution. It can reduce the formation of bubbles. And the second case is that the standard curve shows low signal. As you can see, the maximum OD value of these standards is under 1.0. The cause of this kind of abnormal result is the same as the overall OD value low signal result. We have talked about before in the abnormal result number 1. So I'll just skip this case. Okay, and the last case is high background value. Normally, the blank well value should be less than 1.0 if you use the sandwich ELISA kit. But here in this data, they are all above the line. The reasons for this case can be divided into 2 kinds. One is kit component problem, such as A, substrate degeneration and decreased performance of the plate. If you find the substrate turning blue, please do not use it and contact us for a replacement. And the other kind, of course, is experimental operation problem, listed as B to G. For example, using contaminated wash buffer and concentration of detection antibody or HRP conjugate working solution is too high because of the wrong dilution. Okay, the 3 abnormal results of overall OD and standard curve OD we have talked about are relatively simple problems. Next, we'll see the most complex problem as abnormal result number 4, the abnormal sample OD value. This actually includes many detailed situations, but here we would only talk about 2 typical types. The type number 1, like this result data on the right side, you can see the standards works normal, while the sample shows low signal. For this kind of result, different sample types will lead to different analysis directions. If it is a routine sample like serum or plasma, then this result may be relevant to the sample concentration. Here I listed 3 cases. Case 1, if the sample concentration falls within the kit detection range. The reason could be that sample concentration has reduced because of the improper storage, or the detection accuracy is decreased due to the presence of lipids or thickness in sample. Case 2, the sample itself has a high concentration, which is above the detection range. The low signal may be due to the hook effect. The hook effect is a phenomenon of false negative caused by inappropriate proportion of antigen and antibody. Case 3 If the target protein has low expression in the sample concentration may be below the kit sensitive. In this case, the kit cannot meet the needs of the sample measurements. We are sorry. And if the sample type is non-normal, there are also two cases. Case 1, for unconventional samples such as cell lysate, cerebrospinal fluid, etc., the detection accuracy may be decreased due to the matrix effect. The matrix effect is the interactions between the target protein and other elements in the sample, which may lead to inaccurate results. Case 2, for special samples like recombinant protein or the samples from other species, the antibodies may not be able to recognize the specific antigen, leading to a low signal result. Okay, in view of the above 5 situations, we have summarized types of causes and the corresponding solutions here. The most important thing is to reduce the influence of the hook effect and the matrix effect. It can be solved by diluting the samples. We suggest you to search the literature in advance and then make a pretest to determine suitable dilution ratio for your examples. In our ELISA kit instructions, it suggests dilution factors for some sample types. You can also contact our technical supporting team for the recommended dilution factor for your own samples. In addition, it is recommended to select a high-sensitive kit for the detection of low concentration targets. And for detection of recombinant proteins, please confirm the applicability with our technical support team in advance. Okay, the type number 2 of abnormal sample OD. Please see the sample data here. Is abnormal high sample signal. The analysis direction still depends on the sample type. Here we take the mouse interleukin 6 as an example. I also list 3 typical cases for this analysis. Case 1, if the sample is mouse liver and the detection concentration falls within the detection range, it may be a false positive. Since the liver tissue has high content of endogenous HRP, if the plate washing is insufficient, the residual endogenous HRP will lead to a high OD value. Case number 2, if the sample is cell supernatant from model group, its concentration may be much higher than in the control group. And this result may be solved by diluting the sample. And case 3, if the sample is a routine sample, like serum or plasma, it is made because of the hemolysis or contamination in the sample. In view of these 3 cases, it is principle that we need to ensure that the samples are qualified. So it is suggested to search the literature and determine the optimal dilution ratio of the sample through a pre-experiment. And the samples should be collected and stored correctly to avoid hemolysis, contamination, and freeze-thaw cycles. In addition, we need to avoid introducing other sourced substrates as much as possible. If there is the influence of some endogenous substrate, it can be reduced by washing steps. Okay, the last abnormal result is that the standard or sample duplicate wells vary greatly, which means the precision is more than 10%. We can subdivide this problem into the following three cases. First, there is a great difference between both standard duplicates and sample duplicates. This is most likely related to improper storage of the kit, leading to a poor component performance. Second, only the difference between standard duplicates is large. The possible reason is that the standard working solution is diluted inaccurately, or the standard addition operation is not normative. Third, the standards work normally, but the difference between sample duplicates is large. The most possible cause is that the sample addition operation is not normative. Okay, for the above three situations, we can classify the causes of this kind of abnormal results as improper experimental operation, improper kit storage, and unqualified sample property. In addition to the operation details we have talked a lot before, it is also needed to pay attention to the instrument preheating and plate bottom cleaning before the data reading step. And it is necessary to ensure the uniformity of the test samples and to avoid the hook effect or cross-well contamination caused by the high sample concentration. Okay, so today we have learned about how to analyze and troubleshoot 5 common abnormal results. Here I made a mind map which divided the general causes into 3 aspects: the kit usage, the sample collection and processing, and the experimental operation. We should notice them to improve our ELISA result, like the kit storage, and do not use reagents from other kits, and avoid contamination, and pay attention to the incubation temperature, etc. You can see the map after the presentation.